Okay, so protein structure. Now, protein structure, we're going to divide this into 4 levels. Yeah. You're mostly going to be operating on level 1 and level 2. Yeah. Level 3 and level 4 are very hard to predict and usually have to be determined experimentally. But level 1 is primary structure, and that is the order of amino acids. Because remember, every protein is a chain. It's a chain that has different side chains hanging off of it, but it's one long chain, one long backbone. And so that chain can fold up in different ways, whatever's allowed by the side chains or supported by the side chains. But if you know the primary structure, you can reasonably infer, and pretty accurately, the secondary structure. You'll be right something like 80% of the time when you use the best inference methods. From secondary structures, you can then infer how those secondary structures fold up in 3 dimensions to make an actual protein shape. And the overall protein shape is called a protein fold. So it's not like each little folded piece is a fold. It's like the overall structure is a protein fold. That's a good vocabulary word for you that we will use a lot. Okay, so primary structure, amino acids in a row. Alanine, glycine, valine, aspartic acid. Awesome. Secondary structure is what those are doing, and there are a couple different common secondary structures. So this is like your alpha helix, this is like your beta strand, this is like your beta turn, this is like your 310 helix, stuff like that. We'll get into what this means. I'll show you little pictures, and there are videos linked in the document that you can go and watch. And yeah, they help. I think they help me. So, all right, let's see these secondary structures. So first of all, let's talk about hierarchy. So there's a hierarchy, which is that you imagine your primary structure. Primary structure you can figure out if you only have the sequence of the gene. So primary structure can be inferred with near 100% certainty. I think the only thing you're not certain about is if you have, a couple methionines. It might start at the first one, or it might start at the second one, might even start at the third one. But then it's going to be that open reading frame, the one that you inferred using bioinformatics tools, because that's all biology can do, can follow instructions. Yeah. Secondary structure is going to be roughly predictable based on primary. Tertiary structure is sort of predictable. If your protein is related to other proteins, then its tertiary structure will also be related. Quaternary structure is how the different protein folds actually fit together, how they can kind of dock into each other. So if you have 2 different proteins, this is how they can form up and make one unit. That is, if you know the tertiary structure with certainty, you can make a reasonable prediction of quaternary structure. But if, like in this course, you're starting out with straight up DNA data, you can reasonably infer primary structure. I'll be confident about that. Secondary structure, getting a little ropey. 3-dimensional structure, maybe, but probably not with complete accuracy. You'll get some parts right, but you won't be able to tell the right from the wrong, really. And then quaternary structure, don't even try. Very difficult to infer de novo, like from scratch, starting with just nucleotide data. Yeah. All right, so these are the 4 levels. So this is what we described. Primary structure, amino acids in the order in which they come. Secondary structure, alpha helix, beta sheet. It's basically ways that the amino acids are going to be hydrogen bonded together, which will hold that part of the protein rigid. Things around it may flex, but the hydrogen bonded part is going to stay together. Tertiary structure is the 3-dimensional accumulation of all this secondary structure. Alpha helices, beta strands, all coming together to make a beautiful protein. And quaternary structure, yeah, how proteins fit together, because proteins seldom work on their own. We live in a cell. It's like a society of rules. You've got various regulators, and regulators will often work by binding to a protein in such a way that it either promotes the activity of the protein or locks down and prevents the activity of the protein. And so that's how we turn things on and off, because proteins don't really have on and off switches. You have to basically get in there and I don't know if the term makes any sense to you, but you have to kind of monkey wrench them. You have to, jam something in there so that it physically can't do what it was going to do in order to stop a protein. All right, here's an example. And you notice this one is the one from the title card for the entire class and all my videos. So there you go. This is a random protein. It's called NSP15. It comes from SARS coronavirus. SARS-CoV-2 has one as well. Does that matter to you? Probably not. What it is an example of the first 3 layers of protein structure. So you got your primary structure, which is which amino acids are we talking about? And we're looking at this area right around the red dotted line. And so you can see the red dotted line is this part that is extra in the one virus that's not found in the other. And lo and behold, it makes an extra little loop that sticks out because it's extra. Yeah. But look at the rest of the protein. It is staying very nicely together. So this is 3 different versions of the protein all stacked on top of each other. And you see the yellow and the cyan and the dark blue are right on top of each other because these are the same protein and they have not changed shape very much at all. Because that's how proteins work. They assemble precisely. So these are different versions of it. So primary structure, amino acids in order. Secondary structure. These are the alpha helices. These are the beta strands. These are the secondary structure elements, we call them, which are hydrogen bonded together. So we can see the alpha helix that's coming right before this sort of loop, or actually maybe it's down there is the alpha helix. And we can see some of the beta strands that come right after the loop. And yeah, there they are. And so those are just like examples. And then we got the 3-dimensional structure. So that's the tertiary structure. And so there you are. We're looking at it. What we see here in this protein is we're just looking at the backbone. So we're pretending that the side chains don't exist. And we're just looking at how the backbone twists and how the backbone interacts. Protein secondary structures are generally held together by hydrogen bonding of the backbone with other parts of the backbone. There you go. The side chains mainly have to get out of the way. They have to not interfere, not prevent this hydrogen bonding, and then you can actually make these shapes. Yeah, there it is. There you go. All right. So 3 levels of structure with one particular protein. And then this protein. So there are different ways we can look at it, but this is one way. Here's another way. This is like a space-filling model. So here we're imagining all the side chains are on, and this is how the protein actually looks. So this is one subunit of the protein, but actually you get 3 of these coming together that form the bottom, and you get 3 coming together that form the top, and those 3 and 3 are going to bind to each other to make a little sort of hexagon barrel. And so there you go. You get protein A, B, C is around the back, D, E, and F is around the back. Yeah, that's the quaternary structure. So there are actually 6 of the tertiary structures for this particular protein that come together. So that's what we mean. And then this protein actually interacts with other proteins. I think that would still be called quaternary structure. Yeah, this would be like intramolecular quaternary structure, and that would be extramolecular maybe. I don't know. But yeah, just so you can see kind of the way that we build this up, the little layers that go into it. All right, like I said, those secondary structures are going to be stabilized by hydrogen bonding. Such that and it's particularly going to involve that double bonded oxygen, which is right here. And that's going to be reaching out and generally trying to steal a hydrogen atom from the nitrogen. And so this is like a tug of war between oxygen and nitrogen. That is going to be the typical hydrogen bonding structure, although others are possible. But this is, And so it's going to be about 3 angstroms between these two. You're going to have about 1 angstrom between the nitrogen and its hydrogen. 1 angstrom is like a normal chemical bond length. Or you can think of it as like the normal size of an atom, although, the nucleus is way smaller than that. And so the actual distance covered by the hydrogen bond is going to be about 2 angstroms, whereas the regular covalent bond is going to be about a 1 angstrom bond. There you go. Okay. So, hydrogen bonds work. Let's jump past this. This is a thing we can do in class. All right. So what's going on with proline? Well, proline's weird because you've got an alpha carbon, but instead of having a chain that just goes down, it actually reaches back, and that carbon has attacked and bonded to the nitrogen of the peptide bond. Here's the carbon that's holding on to the oxygen, so that's a double bond. And here's the nitrogen that should have some hydrogens on it, and instead we get this carbon attacking it. That's proline. And that will put a solid, kind of unbreakable bend into a protein. And if you have multiple prolines together, they'll start to make, it's like a curved shape, like a very rigid version of a helix that doesn't even have to be hydrogen bonded together. It's just that there are no other options possible when proline is in the house. So proline really limits the options in terms of what the rest of the protein can do. Strongly limits, so there's basically no flexibility allowed in this bond. And this one we know is only either up or down, but with proline it's usually a cis bond, so the carbons are on the same side. And so really you move from a phi and psi to where you only have one of those angles. Yeah. Now, can you figure out which angle can still rotate? What was the name for the angle between the carbon, the alpha carbon, and the carbon that's attached to the oxygen? I'll see if you can figure that out. Yeah. All right. Okay. A little more about protein secondary structures. Stabilized by hydrogen bonds, the 2 main kinds are going to be an alpha helix and a beta strand. And so we're going to break this section, and then we're going to go a little bit deeper into detail on what's an alpha helix, what's a beta strand, and what are the other things that also occur reproducibly within a protein. There are other parts of the protein that will hydrogen bond or will adopt a conformation, but it's one that you don't see reproducibly. It's not something that's necessarily predictable, and we tend to call that random coil. Random coil is not necessarily flexible. It may be very rigid. It's just not an alpha helix and not a beta strand. So it's the third thing. Yeah. A, B, and C for coil. There you go. So that's the ABCs of protein secondary structure. Now come back next section for the details.